All right, so in this lecture, we're going to talk a little bit more in detail about how action potentials get propagated. We talked about this a little bit at the end of the last lecture. So this is a figure that I showed you in the previous lecture. So again, this is like a zoomed in view of this little area of the axon. So at the beginning part of the axon, what will happen is you have the voltage gated sodium channels and opening. Sodium's going to enter. That's going to cause depolarization to the threshold voltage for now the sodium channels that are over here to open, and that's what's shown here. So now these sodium channels open, sodium comes in, depolarization happens. Now this next area gets depolarized enough that these sodium channels open, et cetera, right? So you have the action potential spreading down the axon. Another thing about action potentials is because you're continuously opening new sodium channels, the effect of the action potential doesn't get diluted as you spread further and further away from the point where it started. So that's different from a graded potential where you just have one event of ions flowing into the cell and then they spread out as they travel through the cell. So in this case, you're constantly bringing in new ions because you're opening new ion channels. Okay, so this leads to the question though, when I showed you the picture, I'm just going to go back here again. So you might be saying, all right, well, sodium is coming in here, right? And I understand how that's going to trigger the opening of these sodium channels here, but why isn't it also triggering the opening of sodium channels back here, right? Aren't these sodium ions that are flowing in going both ways? And the answer is yes, they are going both ways, but they are not going to open these sodium channels back here. And this is because action potentials can't flow backwards. The reason why they can't flow backwards is because of something that I told you about, I think it was 2 lectures ago, or maybe 1 lecture. I don't remember when it was. But remember when I told you about how the sodium channels had 3 different states, right? So they have the activated state, the inactivated state, and the closed state, right? So once the sodium channel opens and then closes again, it takes some time to reset before it can open again. So these sodium channels, sorry to jump all over, but these sodium channels here, the threshold voltage for them to open again has been achieved, but they're not going to open again because they're not ready to open again. So this is why the action potential is only going to flow forwards. It's not going to flow backwards because these sodium channels over here are still resetting themselves and they're not ready to open yet. So this is going to create something called the absolute refractory period. So the absolute refractory period is shown on this graph here as basically this period of time after the sodium channels open and close, where you absolutely, no matter what the membrane potential is, you cannot reopen the sodium channels again. There is also, after that, and you can see that this is longer, something called the relative refractory period. So in the relative refractory period, you can open a sodium channel that has just opened and closed, but it takes a larger than normal stimulus, okay? So it's harder than usual. You need to have actually a bigger depolarization in order to trigger the opening of the sodium channel again. But the absolute refractory period is the reason why action potentials don't travel backwards. They can't. All right. Another thing about conduction of action potentials down neurons, I mentioned before that they are going to be facilitated by myelin. So this is going to be made from, in the peripheral nervous system, Schwann cells, or in the central nervous system, oligodendrocytes. These are both different types of neuroglial cells that make up this myelin. And myelin wraps around the axon, okay? And so you might say, well, isn't that going to make it actually kind of hard for action potentials to happen because action potentials require sodium and potassium passing through the membrane. If you have this thing wrapped around it, right, you can't do that. So yes, that is the case. And that's why myelin actually speeds up the transmission of action potentials. So there are these little gaps between the myelin that are called nodes of Ranvier. So these are the little regions between myelinated areas, and you can actually see these under a microscope if you look at an axon. And it turns out that this process of opening and closing sodium channels, it happens very fast, right, in a fraction of a second, but it does take some time. So the fewer ion channels that you have to open in order to propagate the action potential, the faster it's going to move. So what's going to happen is that the sodium ions that flow in here, rather than having to open a sodium channel right next to them and more channels, what happens is these sodium ions actually flow down the length of the myelinated area inside the axon. and they will depolarize the first sodium channel in the next node of Ranvier. So this is going to very significantly speed transmission because you're just cutting out the number of ion channels that need to open and close. So this really speeds it up by quite a bit. I wanted to mention that there are a group of diseases called demyelinating diseases that affect myelin specifically. The first one you might have heard of that I'm going to discuss, which is multiple sclerosis. This is a fairly common disease. It's an autoimmune disease in which your immune system attacks your own myelin. And because it attacks myelin fairly indiscriminately, there are a lot of different symptoms that can be associated with it. Pretty much anything that your nervous system does can be affected by demyelination. So some of the most prominent symptoms are things like muscle weakness, difficulty with movement, feeling tired, having decreased motor coordination, meaning a diminished ability to move your body in the way that you want to. Sometimes it has the effect of slurred speech or vision problems. Like I said, it's quite common. It affects about 400,000 Americans, but I say here the pathology is diverse. So some people have relatively fewer symptoms and live for a very long time without having significant deficits. In other people, it can be much more aggressive, and it can, in some cases, cause death. It can be difficult to diagnose because there are these diverse symptoms and diverse presentations. So one of the best tests is to look at the cerebrospinal fluid that surrounds the brain and spinal cord, which you can test for anti-myelin antibodies. And the pathology is kind of interesting. More women than men develop multiple sclerosis. Caucasians are at higher risk than other ethnic groups. And I think the most interesting one, to me at least anyway, is that people who live farther from the equator are at pretty drastically increased risk of multiple sclerosis as opposed to people who live close to the equator. And the most prominent hypothesis to kind of explain this weird geographic distribution of multiple sclerosis is that it has something to do with vitamin D. So vitamin D, as you might recall, is produced in response to sunlight on the skin. And obviously, the closer you live to the equator, the more exposure you have to sunlight. And so it's thought that having low vitamin D levels, vitamin D seems to be important for regulating the immune system, including it seems to be important for multiple sclerosis, and perhaps has a protective effect against multiple sclerosis if you live in an area where you get a lot of sun. Another one is Guillain-Barré syndrome. This is less common. It causes paralysis, sometimes temporary paralysis, and is often brought on by a specific illness. So you'll get a virus, and then sometimes in a small percentage of people, their immune system just sort of over-responds and ends up attacking myelin in a more acute manner. And then I just put on here many others. There are a lot of other demyelinating diseases out there. Those are just the two most common that I mentioned. But I think these highlight sort of how important myelin is for the normal functioning of your nervous system. All right, and then finally, I wanted to talk a little bit about how action potentials convey intensity of a signal. So action potentials are always the same magnitude. There's no such thing as a bigger action potential or a smaller action potential, and that's because those threshold voltages for opening and closing of sodium-potassium channels are always the same. So an action potential will always produce the same fluctuation of voltage, okay? So we need some way, though, to determine, if you're touching something, you want to know if it's, just a light touch or if somebody's like hitting your finger with a hammer, right? You need a way to distinguish between that. So how do you do that if action potentials are all or nothing? Well, the answer is that you can vary the frequency of action potentials. So you can see here that in response to various stimuli of different intensities. So here you have a stimulus that is going to be so little that it's actually below the threshold for triggering an action potential. So even though the stimulus is present, maybe something very lightly touching your finger, but you actually don't even detect it because it doesn't hit the threshold for picking up the signal. Then a slightly larger stimulus is shown here, and now it's enough to trigger action potentials, but you trigger them relatively infrequently. And then here you have a much bigger stimulus. It looks like it's about 3 times this initial one, and that will cause you to increase the frequency of action potential. So remember that relative refractory period that we just talked about a couple slides ago? right? If you have a really strong stimulus, that can trigger a neuron to fire multiple action potentials very close together in time. And what's going to happen is that will cause the neuron to respond in a more robust way to whatever it is in response to that higher frequency of action potential. So by varying the frequency of action potentials, the nervous system can encode how strong a stimulus is. And then I finally just wanted to mention some other things that can influence the propagation of action potentials in neurons. So there are a number of drugs in particular that target neurons and neurotransmissions by altering ion channels. So some of them you've probably heard of. So the first one, Novocaine. So that's something that you've probably maybe gotten at the dentist before. So Novocaine is the brand name. Procaine is the generic name. So this is a drug that numbs you because it specifically blocks sodium channels in pain sensing neurons. So pain sensing neurons are neurons that respond when something very damaging happens to your mouth and send a message to your brain. So what happens when your dentist injects you with Novocaine is that you're still experiencing that stimulus, right? The dentist is still doing whatever they are to your mouth, but you can't send the message to the part of your brain that perceives pain to say like, ow, this really hurts, right? So it has this numbing effect. It's not a coincidence that Novocaine is a very similar word to another drug called cocaine. Cocaine has a very similar function, but it targets other neurons. So that's why it doesn't have exactly the same effects as Novocaine. Another drug, is something called tetrodotoxin. You may have heard of tetrodotoxin. It's actually a naturally occurring drug that's made by a type of fish called Japanese pufferfish, or fugu is the name in Japanese. These fugu fish, they accumulate tetrodotoxin in their livers and in their gonads. And this is a very potent neurotoxin. It binds to ion channels and actually it like permanently binds the ion channels and kills the neurons. So it's actually a really deadly poison. And interestingly, so fugu is considered to be something of a delicacy in Japan. But because you have to remove the tissues that contain the tetrodotoxin very carefully, it's very dangerous to eat. I read somewhere that there's a law that the Emperor of Japan is not allowed to eat fugu because of this. I don't know if that's true or not. But a lot of people who are like thrill seekers like to eat fugu anyway. I guess they like to flirt with danger. And every year, some people do actually accidentally get poisoned by tetrodotoxin and die from this. I don't know why people would. I can't imagine. It's hard for me to imagine sushi being that good that it's worth risking your life. But apparently some people think that it is. Okay, and then finally, the last thing I wanted to mention is that another thing that can alter a neuron's ability to fire an action potential, because it's based on the membrane potential, the difference in charge between inside and outside the cell. If you have alterations in the levels of ions, something different from normal in the interstitial fluid. So if you had abnormally low or high potassium or calcium or other ions in your extracellular fluid, that's going to change your resting membrane potential, even if you haven't changed the chemical makeup inside the cell. And that can make it either more or less likely for a neuron to fire an action potential, which can really drastically affect the function of your nervous system. So that's one of the reasons why your kidneys are so important, right? To maintain those stable levels of ions in your extracellular fluid. Because if you had ion imbalance, if you have electrolyte imbalance in your extracellular fluid, that can make it so your neurons either don't fire when they're supposed to, or that they can send signals when they're not supposed to. And that can have really significant consequences because you think about it's going to control all the nervous function of your body, all the muscles in your body. You really don't want that to get messed up. So maintaining stable ion levels in the extracellular fluid is really important.